So from the last time we know that the genetic material is obviously the DNA. So today we will be discussing about the structure of DNA and DNA replication. So when we talk about DNA, there are three components. A pentose sugar, that is your deoxyribose, a phosphoric acid, and nitrogenous bases. So these three components are an essential part of DNA. Now when we talk about nitrogenous bases, again they are of two types, purines and pyrimidines. Purines are adenine and guanine, pyrimidines are cytosine and tyrosine. So when we talk about DNA, it is a double helical structure. The diameter of the two DNA strands is 20 angstrom, and on the inner side you get your nitrogenous bases. That means your adenine, guanine, cytosine and thymine. Now the thing to remember is adenine always pairs with thymine with the help of two hydrogen bonds. Guanine pairs with cytosine with the help of three hydrogen bonds. So this structure of DNA was proposed by Watson and Crick. According to them, DNA is a double helical structure in which the nitrogenous base is on the inner side and each strand runs in a 5' to 3' direction. Both these strands run in the opposite direction. That means one runs in the 5' to 3', the other runs in the 3' to 5', and they are a helical structure. Now the nitrogenous bases, that means adenine is always equal to thymine. By this what it means is purines is also equal to pyrimidines. This is also known as the Chargaff's rule, in which he states that the number of purines is equal to the number of pyrimidines in a DNA molecule. So whenever a cell divides, the genetic material also has to be divided. So this is accomplished with the help of DNA replication. By DNA replication, what it means is DNA is making exact copies of itself. So when we talk about DNA replication, there are of three types. The first one, what happens is both the strands separate out. A new strand that is complementary to it is created, but finally what happens is both the parental strands, they come together. That means these are the parental strands, they are coming together, and a newly synthesized DNA is what you are getting. Since the parental strands remain together, such a type of replication is called conservative replication. The other is semi-conservative replication. In semi-conservative replication, what happens is both the DNA strands, they separate and each synthesizes a new DNA strand. So this new DNA has one parental strand and one newly synthesized strand. Such a type of replication is called semi-conservative replication. And finally, there's the last step of replication in which numerous breaks are created in the DNA. That means each break will synthesize a new strand. This will also synthesize a new strand. So obviously what you'll be getting is this type of DNA. That means a single strand contains both the parental as well as a newly synthesized strand. Such a type of replication is called dispersive replication. So generally when we talk about DNA replication, it means DNA makes new copies of itself. So these are of three types: conservative, semi-conservative and dispersive replication. In conservative replication, the parental strands remain together and a new DNA is created. In semi-conservative, the new DNA contains one parental strand and one newly synthesized strand, whereas in dispersive, a single strand consists of both the parental DNA as well as the newly synthesized strand. So experimentally it has been proved that semi-conservative DNA replication occurs the most, and this proof was provided by Michelson and Stahl. So when we talk about semi-conservative replication, Michelson and Stahl, what they did was they grew two bacterial colonies in separate medium, one containing a heavy isotope of nitrogen 15 and the other containing an isotope of nitrogen 14. So that means when the bacteria were growing in this medium, this bacteria had N15 in its DNA and this bacteria had N14 in its DNA. Now this bacteria was subjected to a centrifugal machine. So because of centrifugal machine, what happens is from the results, the DNA were arranged in separate bands. The higher band represented the N15 and the lower band represented the N14. Now, the same DNA, they were grown together in a neutral medium. Now, both the bacteria, since that was one containing N15 and the other containing N14, were growing in the same media, after a few generations, they were also subjected to a centrifugal machine. Now, in this case, what they got was in the first generation, most of the DNA, they either banded in the N15 region or in the N14 region. But after the second generation onwards, most of the DNA banded in the intermediate region, which shows that both DNA now contained or belonged having N15 or N14. By this, what it means is very simple. Bacterial 1, that means one having a nitrogen 15, bacterial 2 having nitrogen 14. They started to replicate. That means their DNA, they started to separate out. So over time, what happens is one strand contained N15 and the other strand contained N14. So this was the majority type, which shows that the DNA replication was of semi-conservative type. So how DNA replication occurs is, first there is a site of origin, also known as the ORI-C site. This enzyme or this site, let's say it is over here, invites certain proteins or enzymes. The first enzyme that attacks it is the helicase. The helicase first attaches to the ORC site. Then after helicase, primase also acts on this particular site. So what happens is the DNA, it starts to uncoil, creating two separate straight strands. One is running in the 5' to 3' direction, the other running in 5' to 3' direction. Now, when helicase attacks the ORC site, primase also enters it. At the same time, single-stranded binding proteins also enter the site. These proteins, what they do is they keep the DNA strands in a straight matter. Then at the same time, other group of enzymes also called topoisomerases, they come and enter. They also help in preventing the recoiling of this DNA strand. So with the help of a single-stranded binding proteins and topoisomerases, this DNA strand is kept in a straight pattern. Now, DNA replication always proceeds in a 5' to 3' direction. And for this, you require RNA primers. RNA primers are small, short segments of RNA. And these RNA primers are produced by primase. But the primase, it alone is not capable of producing primers. It produces primers only in the presence of helicase. So what happens is an RNA primer is created and DNA replication starts. That means from the 5' to 3' direction. But in this case, only one strand is continuously synthesized, because I already told you DNA replication always proceeds from 5' to 3' direction. So DNA replication does not occur from the 3' to 5' direction. So what we get is small breaks of DNA, and these small breaks of DNA are called Okazaki fragments, discovered by the scientist Okazaki. So you might be thinking, since this DNA is not completely synthesized, if a DNA replicates, then we might be getting only one complete DNA and one DNA having breaks. But this is not the case. What happens is this breaks again later completely joined with the help of ligase enzymes. So as a result, we end up getting two completely synthesized DNA strands. So this is it.